All right, my name is Tyler Jensen,
And I'm Ben Tyre.
and today we're going to be doing our presentation on Ectopia cordis. I believe we've talked about this a little bit in embryology. Dr. Reynolds has gone over it a little bit, but we're going to go over it today a little bit more in depth. But just to recap, Ectopia cordis is characterized by the heart either being partially or completely outside of the thorax. So here you can see an example of a young lady with the heart partially in the thorax, and here, more completely outside of it. And today we're going to talk about, things that can cause this. We're going to talk about how the heart and sternum should normally develop. Because when you have problems with both of those, you can see things like ectopia cordis. After we talk about how it's normally supposed to develop, we'll talk about how what goes wrong, some things that can lead to this. And then finally, we'll conclude with some treatment options and, survivability of an infant who's born with this. So we'll start off with the heart formation.
So the heart formation and development begins early on in the splanchnic mesoderm. There we begin to see the first portions of the first heart field forming, which ultimately leads to different factors such as BMPs, Wnt signaling, NCadherins, FGF-beta, which contribute to the formation of the linear heart tube. Once at this stage, the basic structures of primitive ventricles, primitive atrium, and primitive epicardium are formed. And so at this point, all that's left to be done is to use those signaling pathways to basically loop the heart in a process called cardiac looping, which ultimately leads to the segmentation and formation of these different chambers of the heart. We also have migrating neural crest cells, which lead to branching of the various pulmonary trunks and arteries and veins that allow for blood to be pumped in and out of the heart effectively. Any kind of disruption to these pathways can lead to malformations of the heart, which can lead to malpositioning of the heart as well. If the heart is in a bad position, then it can be the beginning stages of ectopia cordis. So one of the other issues that we can see is malformation of the sternum. But in the sternum, it's normally created in this particular shape in order to allow for greater surface area of protection for the heart and the lungs. As you can see in these costal notches, it's supposed to articulate with these ribs, specifically 2 through 7 in early development, and allow for that kind of protection and also a stabilization or anchoring of the shoulder to the thoracic cavity. So here we see the beginning stages of this sternal development. We have these mesenchymal bars, also known as lateral sternal plates, which are embedded within the anterior body wall. So as those begin to condense, they'll eventually grow ever so slightly enough to be able to start connecting with the ends of these primitive rib formations. Once they connect, the ribs will continue to grow medially towards the center of this embryo's body line. As that medial growth continues to push forward, they're allowed to touch one another and eventually will fuse down that midline around week 8, corresponding with the heart descending down into that thoracic cavity like we talked before. This is usually done through the mesenchymal Wnt signaling that we will talk about in our next slide.
Yes, indeed. So this is a study that we found that kind of talks about how the Wnt signaling is involved in this, and the researchers were able to figure this out by removing a genome that coded for something called Wnt-less. Wnt-less is a G protein coupled receptor involved in the Wnt pathway, and so they were able to remove this gene and kind of see what happened in the developing embryo. So some of the defects they saw were craniofacial, skeletal, and midline defects, including a malformed sternum and an exposed heart. So here in these figures, you can see, we have a normally developed rib cage and sternum in the embryo, whereas here, this is the knockout. We see, the ribs are much smaller, and the sternum is completely absent. So it didn't even fuse. So obviously Wnt is heavily involved in this pathway. We see less rib cage growth, so very heavily involved. Further in the paper, the researchers went on to show how they proved that it was Wnt. They showed how there was beta-catenin in the midline, how there was beta-catenin in the mesenchyme. So they showed that it was Wnt. We didn't include it just for the sake of time. The thing we did want to include from this paper was how Wnt is involved in causing the fusion at the midline. And it's through two things. It helps with apoptosis and with migration of the mesenchyme to the midline, like Ben talked about. So here in these figures, you can see this green that we're seeing right here represents apoptotic cells. And so here in the control, we see, there's proper apoptosis going on in the midline, which allows the two mesenchymal lateral plates that Ben talked about to come together. Here, we don't see any apoptosis. So, it kind of prevents those things from coming together. We also just see a complete lack. Well, not a complete lack. We see less migration. So here, we can see that these plates have come together. Very nice. And here in the knockout, they obviously haven't come together. So that is kind of what Wnt does to further prove that this Wnt pathway is involved in cell migration. They did an in vitro test of the migratory abilities of mesenchymal cells, and basically they found normally these cells were able to migrate a certain amount, but when they had the Wnt knocked out or removed, they were migrating much slower. So they further proved that in this paper. To conclude, they kind of did a suggested pathway that Wnt that they used to form this fusion. So here we can see the epithelial cells, expressing Wnt and causing the mesenchymal cells through these Wnt pathways to make the chondroblasts like fuse and migrate properly. So this is kind of their suggested way. Obviously, we don't know 100% exactly what causes the lack of fusion, something wrong with these Wnt pathways. But the exact etiology of why, we see Ectopia cordis isn't known yet. We just know it's something with the Wnt pathway going wrong, leading to the lack of fusion. Now, going off of that, there's different types of Ectopia cordis. Like we saw earlier, you can either be completely out, you can be partially out. We can also see, if it's just the heart that's out, that's called isolated Ectopia cordis. But you can range from this isolated version to a complete ventral evisceration, where you have liver, guts, lots of other things, leaving the body cavity where it's definitely not supposed to. And so we're going to talk about some of those other things that lead to Ectopia cordis. And some of these, don't necessarily have anything to do with this Wnt pathway. They're mechanical issues or other things leading to Ectopia cordis. The first one is something called the pentology of Pantrel. And pentology means consisting of 5 parts. And so there's 5 parts to this defect. And they are written here: deficiencies anterior diaphragm, midline supraumbilical abdominal wall defects, diaphragmatic pericardium defects, various congenital intracardiac abnormalities, and a defect of the lower sternum. So there's these 5 things that, make up this pentology of Pantrel. And once again, the etiology of this isn't 100% known. They don't know exactly why this happens. Probably something to do with these Wnt pathways, maybe something else leading or making it so these, organs are leaving the body and aren't being contained like they're supposed to be, So this is the first one. The next one is something called early amnion rupture. So here you see, here you see the embryo, and it's surrounded by the amniotic cavity. And sometimes this amniotic cavity can burst. And this leads to different pressures than the embryo is used to, maybe by the yolk sac or just the yolk itself pressing on the embryo in a way it's not supposed to. So this leads to mechanical pressure of the heart, which interferes with the cardiac descent that Ben talked about earlier. And so if the heart can't descend, then it's in the way of the sternum, and that can't form properly. So this is one of the possible etiologies for Ectopia cordis. Not every time there's amniotic rupture, we see Ectopia cordis. That's just occasionally, amnion rupture, sometimes you see it, sometimes you don't. It's just possibly, an influencing factor. Also, sometimes when you see amnion rupture, it also leads to something called amniotic band syndrome. So that's here. So if you look up here in this figure, you can see that there's these fibrous bands in the amnion. And when it ruptures, these bands can get wrapped around the fetus. So here you can see it wrapped around the fetus that'll probably lead to some limb deformity. These bands can get wrapped around the heart and also limit its ability to descend. So here you see the heart, we have this arrow pointing here to this band. This band also caused, some facial deformities. So this is another example of a mechanical thing leading to Ectopia cordis. This is embryologically different from isolated Ectopia cordis. This isn't really anything to do with the Wnt pathway. There's other things involved here different than the Wnt pathway. These are just some other things that can lead to it. The final one is something called trisomy 18, which is problems with meiotic division, where the fetus will end up with an extra copy of a chromosome. And this leads to a ton of deformalities. The fetus is usually aborted. These deformalities include cardiovascular, nervous, renal, gastrointestinal, respiratory, and musculoskeletal system. So a ton of problems. And here's a picture of the aborted fetus. Obviously, you can see this heart, is outside of the cavity, not where it's supposed to be. Studies have shown that after the age of 45, women that bear children, the chances of them having trisomy 18 are increased. So just those are a couple of the things that can lead to Ectopia cordis. Some to do with the Wnt signaling, Wnt pathway, some to do, with just mechanical things that led to the heart not forming properly. So now we want to conclude by talking about some of the treatment options, and Ben will take it from here.
Yeah. So as you can see with the majority of these things that Tyler's talked about with these deformities, treatment outlook is usually not that good. Our prognosis is pretty bad in most cases. However, in some cases, there is a good surgical outlook or even an ability for people to live a normal life without any surgical intervention, such as with this girl who happens to have a good coverage of the heart with skin and enough room for the heart to continue to function normally within the thoracic cavity. If we have a salvageable pericardium, sufficient space in order to reduce that heart back into the thoracic cavity, and then limited additional congenital heart defects, such as omphalocele and other issues with the heart that would render it irreparable, there is a good prognosis. The main reason for this being that if we can put the heart back into the chest, that still needs to be able to function properly once it's there. Mm-hmm. If there's no way to fix it while it's in there, then there's no way for the baby to have a good prognosis later on. Mm-hmm. So prognoses are also improved when they're detected earlier on in pregnancy. It can be detected as early as the second trimester using 3D ultrasounds. And that basically just helps us to understand how the birth process can be made easier to prevent further damage or to determine whether or not the baby needs to be aborted earlier on in order to save the mother. But in most cases, we're going to want to perform these surgeries earlier on, preferably before the 3-month mark of the baby, when the thoracic cavity becomes too rigid to use already existing structures within that thoracic cavity, such as the ribs and what is remaining of the sternal plate. So along with that, there are multiple different localizations of these deformalities. Specifically, there's cervical, where the heart is growing up more into the neck area, which is a lower prognosis for being able to be surgically repaired. Then there's also thoracoabdominal and thoracic types of Ectopia cordis, such as in this girl, where the repairability is a much higher outlook. And so with the treatment of, with the surgical treatment that we can do in some cases, all we really need is to be able to provide a soft tissue to cover the heart, to reduce the heart into the thoracic cavity, and to repair any of the intracardiac defects to reconstruct the chest wall. And so we're gonna let these guys explain what they did here.
I thought of was a way to use some existing bone and cartilage in the area to move it over to the area that needs to be protected. And to do that, we would just sacrifice areas out where we didn't need the protection as well. So we really used some of the adjacent bone and cartilage to cover the heart so that he'd have stable coverage.
Pretty cool.
And that's our presentation.
I thought it was a way to use some existing.
So that's our presentation.